Good evening, everyone. So, welcome you all to week 2 NPTEL TA session on RNA as enzymes. Okay, so this is Subhasmita from IIT Indore. I am a PhD scholar here. So, let's start today's class. So, last class we have discussed few things. People are joining, so I need to admit them. Let me know if my voice is not clear to you, if I am not audible, or if my screen is not visible to you, then please let me know in the chat box. Okay. So this session is overall about our RNA biology and RNA world hypothesis. Okay, so in the previous form of life origin, it started from RNA. Okay, it is believed there are few molecules, macromolecules, that is RNA, DNA, protein, which is majorly found inside a living system, and from this it is believed that RNA was the first macromolecule to be found in the preliminary era of life origin. Okay, and RNA is made up of ribose sugar and phosphoric acid and anyone nitrogenous base. Okay, so previously we have discussed few things about RNA world hypothesis. So I want to just recall it again and discuss it few again times. So let's talk about it. So what is RNA world hypothesis? It was that first discovery of catalytic RNAs or ribozymes. Okay, so this catalytic RNA or ribozymes, they are basically RNA acting as enzymes. Ribozymes are called ribonucleoproteins. That means there must be one RNA molecule and one protein molecule make a complex. Okay, so in this RNA molecule act as enzyme, not protein. Generally, in our body and all living system, we can find that protein is acting as enzyme. Tell me any enzyme, you will see that it will be a protein, not RNA. But in the previous world or the beginning of life, it has been found this enzyme, that is ribozyme, where RNA acts as a catalyst, biocatalyst. Okay. So next thing, it is believed in the figure also you can see, just a minute. Okay, so here it is believed that RNA first, that is RNA world hypothesis. It says RNA chemistry. So this is believed to be RNA along with protein complex, that is ribonucleoprotein or ribozyme, which leads to molecular self-replication. That means at that time a ribozymes replicated itself. Nowadays RNA cannot replicate itself. It replicates using a template with different presence of different enzymes. Okay, so molecular self replication has been happened for ribozymes, that led to a uh, prior organism or prior cell, we can call it as a protocell. Okay, so this is the outer layer of a protocell and this is the inner membrane along with some hereditary molecule, that is believed to be RNA. Okay, the molecule which has the power of inheritance, that transfer from one parents to one generation to other generation. Okay, and the second one is discoveries of ribosomes. So these are few points which says us that yes, it is the molecule RNA which was found in the previous form of life, not DNA or protein. Second one is discoveries of ribosomes. So ribosomes are large ribonucleoproteins and they translate RNA into protein. So in the translation process, what happens? This mRNA molecule, that is mature mRNA molecule. We will be talking about mature and premature mRNA. So mature mRNA molecule, it acts as a template, and using this mature mRNA long chain, this ribosome translate it into a protein. Okay, and this protein can act as different forms of, can be acting as a form of structure building, can be act as a form of enzyme or anything else. Okay, so next thing is, third supportive research demonstrated artificially constructed RNA molecules can catalyze almost any imaginable reaction needed in a living system. And lastly the scientist, what they did, they just made RNA molecules, artificially RNA molecules, and they try to understand if it can catalyze the metabolism process inside our body or not. And it has been found that yes, it can catalyze different metabolic processes. Okay, so now it is clear that yes, RNA can self replicate itself in the previous era, right? Next, finally and most recently, RNA sequences capable of simple forms of self replication have been also discovered. Some sequences of RNA has been believed to be capable of self replication also. Okay, so this few points made us believe that RNA was the first molecule present in the beginning of life, not DNA or protein. Okay, so there are few these are the few points which made us clear about RNA world hypothesis. Okay, I guess we have just a brief discussion about the RNA world hypothesis. So let's move to our question answer session. So this is the first question of today's session. Which one of the following is believed to be a remnant of RNA world? Self-splicing exons, self-cleaving viral RNA, or RNA polymerase, or none? Which one it will be? Please try to answer this question. Write your answers in the chat box. Now we have discussed about RNA world, so I believe everyone can answer this question. Please try to answer. If you don't know the particular answer, then you can also guess it. We'll discuss about it. No worries. Only some of you have answered. Everyone, please try to answer. Others, please try. Okay, then let's discuss about it. Okay. So here your answer is self-cleaving viral RNA. Some of you have answered C, RNA polymerase. See, RNA polymerase is a protein. You know this? RNA polymerase is an enzyme where protein acts as an enzyme. So, if it need to be a remnant of or remaining of RNA world, that means it needs to be one thing that is made up of RNA, not protein. Okay, self splicing exons. So here it is removed, that is self splicing exons. Next is exons are what? Can anyone tell me what are exons? What is the meaning of exons? Yes, you can guess it. Just write it down in the chat box. Yes, very good. So here it is the coding RNA. We can call it as the coding sequence, right? Thank you. Thank you for answering. So these exons are basically coding RNA. So self-splicing, why we do need to just splice the exon? We need to splice the introns. So here this option is removed by itself only. Next is self-cleaving viral RNA. Okay. Self-cleaving, we'll talk about it. Next is RNA polymerase. RNA polymerase is made up of protein, so it will not be a remnant of RNA world. Okay, none is less. So here you can see what are the possible remnants of RNA world. Understood? So that is RNSP, that is ribonuclease P ribozyme that cleaves tRNA precursors. Understood? We will be talking about ribonuclease P in a few upcoming slides. Next is your self-splicing introns. Yes, we need to splice the introns, not the exons. Okay. Next is self-cleaving viral RNA. So basically what happened in some of the cases, in some viruses, the genetic material is RNA, not DNA. In higher living organisms that like human and other mammals, the hereditary material, that the inheritance material is DNA. But in case of some viruses, the genetic material is RNA. So this self-cleaving viral RNAs are one good example of how this RNA was the first molecule, not DNA or protein. Okay. Next is peptidyl transferase in the ribosome. So this is also one enzyme responsible for translation process. We will be talking about it. Next is nucleotides involved in metabolism and signaling. Of course, nucleotides are involved in all different types of metabolism and different types of cell signaling processes. If you have heard about any type of signaling, there must be one receptor and there must be some molecules which factors which binds to that receptor leads to the complex formation followed by a cascade of process that leads to cell signaling, and at the end the work is done. Okay, so obviously nucleotides have been included in lot of signaling processes and metabolisms also. Okay, Anuya, please tell your friend to join again. I'll admit it. Okay, it's done. I guess it's clear to everyone. Okay, so this is the possible remnants of RNA world. Okay, so now this is a very simple question. So please try to answer it. RNA can replicate, form protein, form DNA, act as a biocatalyst. Which one is true? See, we have discussed it a lot of times, so I believe most of you can answer it. Everyone, please try to answer. If you want to ask any question, then you can write it in the chat box. Okay, so let's discuss. So here I got a diverse of answers. Some of you have said B, some of you have said D, some C and A. Okay, so every options have been covered. Nice. So your answer is D. Okay. RNA can act as a biocatalyst, which we have been talking about from last 10 minutes. Okay, RNA can act as a biocatalyst in the form of ribozymes. In the ribozymes, there is one part is protein, one part is RNA, where RNA acts as the enzyme, not protein. Okay, that's why RNA is called a this ribozyme is called a ribonucleoprotein, and here RNA act as a biocatalyst. Fine. Okay, all seems to be correct. See, RNA cannot replicate, only DNA replicate. Okay? So here we'll be discussing these things. Just be with me. Uh, RNA can replicate. Only DNA can replicate, not RNA. See? And next is RNA can form protein. It directly cannot form protein because mRNA acts as a template. In that case, mRNA just act as a template, and lot of machineries like using tRNA and other enzymes, they require it require a lot of enzymes to make a particular sequence of primary sequence of amino acid, that is the simple primary protein. Okay, next is form DNA. RNA cannot form DNA, but DNA cannot form can form RNA. Okay, so this option can be true in case of some viruses. Like, can anyone tell me RNA can form DNA or not? Where? Where? which case RNA and what is the process called?
Reverse transcription.
Okay, very good. But right, yeah, I understand it. Okay, thank you for answering. Just write it down because if everyone just talks simultaneously, it will be chaos. Okay, I understand your concern. Okay, so here it will be also C, that is DNA. Okay, you got it right that it seems quite complicated, but yes, it can form DNA in the form of reverse transcriptase process. Okay, if which is present in basically which virus? Very simple virus, everyone knows, HIV. Very good. So in case of HIV, we mostly seen this process reverse transcription, that is RNA forms the DNA. Okay, and we can relate it these two options also, but there are a lot of machineries required for it, so it can't be called directly to protein or RNA can replicate directly. Okay, ribozymes can replicate, but RNA cannot replicate. See, let's talk about it more. So next thing is ribozyme. This is ribonucleic acid and enzyme, and it is not a protein. In 1989, Nobel Prize in Chemistry has been awarded to Sidney Altman and Thomas Case for the discovery that RNA in living cells is not only a molecule of heredity but also can function as biocatalyst. Okay, these two scientists got the Nobel Prize in 1989 for the discovery of this molecule of heredity, that is RNA, which can function as a biocatalyst. Okay, so let's move to next question. Ribozyme can increase the rate of metabolism, transcription, translation, or replication. So ribozyme we have talked about, it has one RNA part, it has one protein part. That's why it is called a ribonucleoprotein. So now tell me, it can increase the rate of metabolism, transcription, translation, or replication. Okay, so most of you have answered C, some of you have answered B, right? So let's see what is the answer. So this is one slide from the lecture notes. You can find these slides from your NPTEL folder also. So let's talk about ribozyme a little bit. Ribozyme is the RNA possessing catalytic activity. Okay. Next, increases the rate and specificity of phosphodiester bond cleavage and peptide bond synthesis. So, if you see it correctly, in the translation process, peptide bond formation is required, right? One amino acid is there, another amino acid is there. There needs to be a peptide bond. Then the chain continues, and that leads to a primary sequence of protein. Okay, primary protein, primary structure of protein. But in case of phosphodiester bond cleavage, if any process where this RNA needs to be broke down, that's when we can use this ribozyme, which will help us to break the bond, that is phosphodiester bond cleavage. In the process of transcription, generally what happens? DNA to RNA, that means RNA has been made, right? So in the process of RNA making process, there needs to be the synthesis of phosphodiester bond, not cleavage. Okay, there need to be a phosphodiester bond between two nucleotides so that the RNA chain can move move away, like increase its length. Okay, so here, as you can see, so your answer will be translation. Understood? Because in case of transcription process, the phosphodiester bond needs to be made, not broken. Okay, we need to make the phosphodiester bond, synthesize it. That's why we'll go for translation, where it will help us to synthesize the peptide bond. Okay, and a widespread occurrence of nature has been done by this ribozyme from viruses to humans. Okay, so in humans and in also in case of viruses also it has been found. I guess it's clear. So let's move to next question. Which ribozyme helps in maturation of tRNA? So it has been found that there are a lot of ribozymes. So which ribozyme helps in maturation of tRNA? rRNA, hairpin ribozyme, self-splicing ribozyme, or ribonuclease P ribozyme? Everyone, please try to answer. If you don't know the answer, then also you can guess it. We will be discussing it in a few minutes. Okay then, so let's discuss about it. So some of you have answered rRNA, hairpin or ribonucleus P. Okay. So see, there are few naturally occurring ribozymes, and here it has been listed that which ribozyme is helping in which catalytic function and the biological function. So this rRNA and tRNA, they help in peptide bond formation. So this will be the biological function of protein synthesis or translation. Okay, you can just remember it by their function. Next is ribonucleus P. Here it helps in phosphodiester bond hydrolysis. Okay, phosphodiester bond hydrolysis. Hydrolysis means breaking of phosphodiester bond in the presence of water. Hydro means water. Lysis means breakage. So breaking of phosphodiester bond in the presence of water is called phosphodiester bond hydrolysis, which here the biological function is maturation of transfer RNA, that is tRNA. So which ribozyme is required for it? That is ribonucleus P. Your answer, right? Next is self-splicing ribozyme. What is its function? Next, same thing, phosphodiester bond hydrolysis and ligation. So ligation, what is ligation? Generally, it has been found during your uh, nucleotide, that is your replication and transcription process. Okay, this ligation process just make the synthesize the phosphodiester bond between two nucleotides, which remove introns from precursor messenger RNA. So here precursor messenger RNA, it has both introns and exons, okay, which has been just transcribed from DNA. It has both introns and exons, but in the splicing method, the introns have been removed, only the exons remain. Okay, so for that process, ligation is required. Okay, so that precursor messenger RNA becomes mature mRNA after the splicing process. Understood? Next is this splicing ribozyme. It can be of group 1 or group 2 introns. Next is your hairpin. Hairpin ribozyme. It helps in, or hammerhead ribozyme or HTB ribozyme. This helps in phosphodiester bond hydrolysis, the same thing. And this is required for your RNA virus replication pathway. So we'll be talking about HTB, hammerhead and hairpin ribozymes in upcoming slides, that how it is helping the RNA virus replication pathway. RNA virus means the genetic material of this virus is RNA. So now you got your answer, that is ribonucleus P. You can just remember this, or I think it's easy because you know every ribosome has its specific function, and we need to understand, if you want to understand the mechanism of RNA biology, then it will be helpful to remember it, so that it will be helpful in understanding the things. Next question, do the catalytic strategies of ribozymes and proteins are similar? Yes or no? Everyone knows here that ribosome has a power of biocatalyst, right? So it can catalyze the process. Next, proteins also act as enzymes in lot of metabolic processes. So do the catalytic strategies or catalytic process by ribosome or proteins are similar or not? What do you think? There are two options, yes or no. We will discuss it. So just try to answer it after thinking a little bit. What will you think? Okay, so here I got a lot of answers saying A, that is yes. You all think that it's yes. That means the catalytic strategies of ribozymes and proteins are similar. So let's see. Okay, appear to be similar. It's true. RNA as well as protein enzymes use acid-base groups and metal ions to activate nucleophiles and to stabilize developing charge on the leaving group. Okay, so this is the whole process of catalysis if you find it. Okay, generally acid base groups have been used in case of protein where this acid base group is present in the R group. Okay, this amino acid has four bonds, right? In the center is carbon, one side it will be hydrogen, other side it will be your R group. Next two sides, one is amino group NH2, other one is COH, carboxylic acid groups. So the R group is essential for the acid base nature of this particular amino acid. Okay, it changes from one amino acid to other amino acid. Also the metal ions, if you can remember, the lot of metal ions including manganese and calcium and others, that metal ions have been used for a lot of metabolic processes. Generally kinase mediated this regulation processes have been there, lot of metal ions have been used. Also in case of your hemoglobin also there is specifically iron is present, right? So this act to activate nucleophiles and stabilize developing charge on the leaving group, the same process have been used both by RNA and protein enzyme. Next is ribozyme also requires formation of a specific secondary and tertiary structure of RNA by base pairing of complementary regions. Specific primary structure of certain regions is also necessary. So ribozyme, generally protein has four structures, right? One is your primary, secondary, tertiary and quaternary, right? So in these four structures, these secondary and tertiary structures are specifically required for the enzymatic action. The primary structure of a protein is a simple chain of amino acids. So it generally doesn't act as enzyme in an efficient way. Okay, so this secondary and tertiary structure is required for it. Same for RNA also. If you see, the secondary and tertiary structure of RNA is required for the process of catalysis. Some cases primary structure is also required, but secondary and tertiary structure are more prominent. And some ribozymes can speed up the rate of reaction to 10 to the power 3 to 10 to the power 11 times. So in case of proteins also, when we just recall that enzymes are specifically used to faster the process, right? Everyone heard about it in your 12th class also, that enzymes are specifically used to faster the reaction, to make the reaction faster. So same process for ribozymes also, they can also speed up the reaction up to 10 to the power 3 to 11 times. And in case of HTB ribozyme, this cleaves the phosphodiester bond as fast as RNase. So RNase is a protein or RNA? Can anyone tell me? RNase is a protein or RNA? What do you think? Here it is written RNase, right? Yes, can anyone tell me? Enzyme. Okay. So RNA is an enzyme. I am asking if RNase is a protein or DNA, RNA. What do you think? Yes, it's a protein. Very good. Okay, it's a protein, not RNA. In case of HTB ribozyme, it is RNA, RNA plus protein. But here, RNase is a protein, not a RNA. Fine. Now you got your answer also. I guess this discussion is helpful to understand the catalytic mechanism of ribozyme and protein. Okay, let's see. Another question. Which one is not an example of small ribozymes? There are different class of ribozymes. So select one which is not a example of small ribozyme. Hairpin, hammerhead, HTB, RNSP. Okay, so let's discuss. So there are basically two class of ribozymes, that is metalloribozyme and small ribozymes. This metalloribozymes includes ribonuclease P and self-splicing introns, while some small ribozymes include hammerhead, hairpin and hepatitis delta virus ribozyme, that is HTB. So here if you see, your answer is RNSP or ribonuclease P. So this is basically a class of metalloribozyme. So metalloribozyme means there need to be one metal compound to act in the function of this particular ribonuclease P. Understood? So we'll be talking about metalloribozymes. Let's see another question. So here it is. What is the role of metals in ribozyme? Now you just answered one question where ribonuclease P is an example of metalloribozyme. So now the question is, what is the role of metals in ribozyme? It act as catalyst, make the process faster. That means the reaction process faster, or stabilize the structure, or it just remains silent, nothing else. Very good, you are going in a right direction. Everyone else, please try to answer. Okay, so let's discuss about it. So most of you have answered C. Very good. That means you are getting the point here. It just stabilizes the structure because RNA acts as the catalyst. You know that, right? The role of metal ion in RNA catalyst. Ribozymes are generally metal enzymes. For example, your RNAsP and anyone else? We just talked about it. That is self-splicing introns, right? So, but in most catalytic ribozymes, metal do not participate in the catalytic process. They just don't participate in the catalysis, but stabilize the ribosome structure, which have been seen. This is the example where magnesium and cobalt just stabilize the ribozyme structure. Okay, and RNSP just catalyze the site-specific hydrolysis of precursor tRNA, which is essential for the formation of mature tRNA. And catalytic activity depends upon the presence of divalent cations, for example, your magnesium and manganese. Large ribozyme composed of both RNA and protein. However, the RNA moiety alone is the catalyst, which I was saying that RNA only acts as the catalyst, not the metal ion. Metal ion, for example, your magnesium, manganese, they all are required for your stabilization of the process in the process that is ribozyme. Okay. So just can anyone give me one example of using magnesium ion? Do you recall one example of any glycolysis process or any like any respiration process where magnesium ion is used? Anyone? Do you recall it? Okay, so basically magnesium is required by your kinase, right? So kinase का काम क्या होता है? This kinase actually helps in the phosphorylation process, and it phosphorylates and activates the kinase, activates the receptor. So that's why it requires one divalent cation, that is magnesium. Understood? And there are a lot of processes inside our body where generally this magnesium and manganese have been has been required. Okay, that's why we should have magnesium, which is a trace element inside our diet. Okay, sometimes if you must have heard about some people having magnesium deficiency also, and that also affects different metabolic processes inside our body, right? Because magnesium is required for a lot of metabolic reactions. Understood? Fine. So let's talk about ribozymes again. Cofactors in ribozymes are important for. So earlier we have been talking about the metal ions. So here we are talking about cofactors are important for RNA primary structure, structural stability, RNA secondary structure, or RNA DNA interaction. Yes, everyone, please try to answer this question. Can you see the screen? Okay, thank you. Okay, let's discuss. So all of you or most of you have answered C, right? So yes, it's true. So it is required for RNA secondary structure, right? So cofactors in ribozymes. So first thing we need to understand what is a cofactor. So cofactors are basically non-protein compounds. They can be a chemical compound other than protein or can be a metal ion also. So generally they are required in the for RNA secondary structure formation or understood or stabilization of RNA secondary structure. So now the question may arise that cofactors earlier we have just discussed that metal ions stabilize the ribozymes, right? So now it has been the answer that RNA secondary structure is requires this cofactors. So your explanation is this metal ions can be a cofactor, but not all cofactors are metal ions. Okay. So cofactors means non-protein compounds which can be any compound other than metal ions also. But metal ions can act as a cofactor. Understood? So metal ion can act as a cofactor and stabilize the ribozyme. But this RNA secondary structure formation cofactors are required. RNA transcripts fold into secondary structure via intricate patterns of base pairing. This secondary structure impact catalytic ligand binding scaffolding functions to wide array of RNAs, forming a critical node of biological regulation. Okay, so this secondary structure of RNA is important for catalytic process, ligand binding and scaffolding functions also. Here one figure is also there. One is, this is the cofactor molecule. This is cofactor dependent ribozyme that leads to cofactor dependent enzyme ancestor, which leads to cofactor dependent enzyme A. Okay. Next, non-cofactor function. that leads to gene duplication or mutation of some in some ribozymes or some proteins also. Okay, so this is basically a part of some experiment. This is not the whole experiment. I am just showing you the few part of one experiment. That's why this cofactor molecule is required for the secondary structure of RNA. Understood? So let's look into another question. A pre-tRNA molecule is matured by. I guess we have discussed this question before. If you recall your ribozymes, then you can answer it. I believe everyone can answer, so please try to answer. Okay, so here the answer is RNSP action. Okay, we have discussed it before, so I am just revising it again. The RNA subunit of ribonuclease P, because it has one RNA subunit and one protein subunit, was one of the first two ribozymes to be discovered. RNSP catalyzes the process of precursors to all kind of tRNAs, that is pre-tRNAs. The enzyme cleaves the pre-tRNAs, removing 5 days precursor specific sequences and generating mature 5 days ends to the tRNAs. So here one is RNSP recognition, it has been showing. This is the secondary structure. If you see the structure of tRNA, it is basically the secondary structure of RNA because it looks like a clover leaf, and if you just recall about it, it has base pairing between two strands of tRNA, right? Just remember, try to remind the structure of tRNA. It has two strands been base paired, that is the secondary structure of tRNA. So this, maybe it look like this ribonuclease pre in the presence of magnesium. So here this magnesium acting as a cofactor, this actually removes the 5 days precursor specific sequences, generating the mature 5 days ends. Okay, after removing the process of hydrolysis, this OH group have been removed, and it shows a addition of phosphate group, right? So this 5 days precursor have been removed, and now we can call it as a mature tRNA. Actually this process is quite long from pre-tRNA to mature tRNA, but I don't want you to be confused about the whole process, because we don't need to go deeply into the maturation of tRNA. So here this is just a brief explanation how this RNSP is helping in maturation of tRNA, because there are a few number of steps, and each steps need to be specified by different enzymes also. If you want to know about the each and every step of maturation, then you can go for different articles and different sites also. Okay, so here let's move to next question. Central dogma in biology says DNA replicates, RNA replicates, protein replicates, or ribosome replicates. It is quite simple. Others, please try to answer. I only see few people are answering the questions because I believe this is quite simple question to everyone answer. Everyone can answer. Okay, so let's discuss it. As you have answered, this is your answer, that is DNA replicates. So what is central dogma of molecular biology? This is basically your replication of DNA, that DNA replicates to form another DNA in a semi-conservative manner, right? And for this, DNA polymerase is the enzyme which is required. And DNA polymerase, which is the enzyme, this is made up of protein. Okay. Next is your, and this can be also reverse transcription. This reverse transcription process where RNA leads to production of DNA in some viruses. This process, this requires one enzyme, that is reverse transcriptase. This is also a enzyme made up of protein. Okay, so this is the exception of biology where one thing is that DNA to RNA then to protein, but next it has been seen in case of some viruses that RNA can lead to DNA. Okay, so next is DNA to RNA, that is the process of transcription require the enzyme RNA polymerase. Understood? So these are two types of RNA, that is plus sense RNA and minus sense RNA. If you look into the RNA maturation in a quite clearly, then you will find the plus sense RNA and minus sense RNA in case of some bacteria and viruses also. So next, that leads to translation, which leads to protein formation using ribosome and different machineries. And in case of bacteria and viruses, it has been also found this RNA replicates to plus sense RNA to negative sense RNA, where and it requires one enzyme, that is RNA dependent RNA polymerase, that is RDRP. And most of the cases, what is the plus point of this RDRP, which is found in different prokaryotes? Most of the drugs have been designed to target this RNA dependent RNA polymerase so that it won't affect the host body, but it will affect the prokaryotic infecting organism. Okay, that's why RDRP is quite essential and quite beneficial for developing a drug against such viruses and bacteria. Okay, that's the plus point of having RDRP. Understood? Same thing again for your some bacteria and viruses. They contain the amino acids, right? We have also the amino acids, but the L and the clockwise rotation of amino acid and anti-clockwise rotation of amino acids, that is the RNA structure, if you recall it, that is the you can call it the chirality. Okay, chirality of amino acids. This RNA structure is different from the human, that is complex living organism and prokaryotic organism. So if we target the particular structure which has been found only the prokaryotic system, then it will be beneficial to the host body, that it won't affect any amino acid or anything else in the host body, but it will kill the virus or bacteria. Okay, so that's the plus point of having some differences where we will won't have any side effects because of this particular medicine or drug. Fine. Okay, that's the thing about little bit about drugs and drug discovery. So here another question: only bacteria and plants contain the ribosomes. Is it true or false? Only bacteria and plants contain ribosomes. Others, please try to answer. I got only few answers. Okay, fine, then let's discuss about it. So this is false. All of you have answered A, that is true. So how come you thought about that only bacteria and plants contain ribosomes? Don't the animals contain ribosomes? Animals also contain ribozymes, and in case of viruses also we find the ribozymes which helps in the RNA mediated viruses, which helps in the replication process. We just talked about it, right? So just read the question carefully, think about it, then try to answer. Okay, so here it will be false because viruses also has ribozymes. Ribozymes are found in ribosome where they join amino acids together to form protein chains and also play in other vital reactions such as RNA splicing, transfer of RNA, biosynthesis, and viral replication. So which ribozyme is especially helpful in viral replication? That HTV ribozyme, which is also called a small ribozyme, right? This small ribozyme, HTV, hepatitis delta virus ribozyme, is helps in viral replication process. Understood? So it also found in case of viruses also in animals also, not only plants and your bacteria. It says only. Okay. So whenever you try to answer the question in case of your GATE exam or CSR NET exam and other exams, and you find this type of questions, so please, you think that yes, it's quite very easy, we can answer it in a second, but then must be some reason behind the question. So think about it little bit. Understood? Fine. So let's move to next question. The chemical properties of RNA is different from DNA. Is it true or false? Try to answer this question. Chemical properties of RNA is different from DNA. Okay. So here some of you have said true, some of you have said false. Those who have said false, see, the chemical properties of RNA is obviously different from DNA. It is true. Why? Because RNA has this ribose sugar and DNA has deoxyribose sugar. So the presence of OH group in RNA makes it more reactive as comparison to DNA. Also, there are few factors like here you can see DNA versus RNA. That's why I put this question to understand, to make you understand the chemical property difference in chemical property between DNA and RNA. So you can see here deoxyribonucleic acid. Fine. This is deoxy. One OH group is absent here. See here is this is H. This is OH. Understood? So one hydroxy group is absent here. That's why it makes it less reactive and more stable. Understood? And in case of ribonucleic acid, usually single stranded is present, and this ribose sugar also there. Also there is one uh, nucleotide, that is uracil, is present in case of RNA, while in case of DNA thymine is present. So one nitrogenous base difference is there. Next, one OH group difference is there, that is the hydroxy group. So that's the difference between DNA and RNA, basic difference. There are other differences also, you can find it. These are the basic differences between DNA and RNA, which make it different in their chemical properties. Okay, if RNA was not that much reactive, then it would be act as a genetic material in case of higher plants and higher animals also, higher organisms also. But it's not. It cause it acts as a catalyst in most of the cases, and it highly unstable. It leads to mutation in high easily manner. So if this our genetic material is highly prone to mutation, then it will change a lot of things, change a lot of metabolic reaction inside your body, and that is not good, it is harmful, right? So this DNA versus RNA, there are difference in their chemical properties. Understood? Next question. Every RNA has a secondary structure. Is it true or false? Every RNA. You know there are different types of RNAs. Okay, so let's discuss. So most of you have answered A. That is true. So let me explain one thing first. Every RNA, it is said every RNA. Every RNA means there are generally there are a lot of RNAs are present, but generally there are 3 types of RNA, right? mRNA, rRNA, and tRNA. And we know that tRNA is shaped as a clover leaf, and it has a secondary structure where this base pairing happens, right? So now let's talk about mRNA. Have you seen this mRNA in a secondary structure? If the mRNA becomes, see, for example, this is your mRNA, right? This is your 5' end, this is your 3' end, and these are different base has been attached here. Okay, so this is this mRNA. Okay, so this mRNA, if it remains like this, then only the translation process can happen, right or not? So if this remains in a straight line, in a single line, then only the translation process will happen, where this codes will be read by the anticodon of this tRNA molecule, right? tRNA, it has the structure where the tRNA, you, I guess we have this, right? Yes. So here this is the anticodon part of tRNA, where this mRNA molecule, if this mRNA is like this, your mRNA, if your mRNA is like this, then it will be read like AUZ or GAG or whatever it is, the whatever the 3-letter code is there, and in the acceptor arm there will be this amino acid. Okay, I am writing it as AA, amino acid. So if for example the code is AUG, that is the amino acid will come as methionine, like this. Okay, so if the mRNA becomes a secondary structure like this, then it can't be read. Don't you think that if this mRNA structure becomes changes, that it changes to a secondary structure, so how will the tRNA will read the code? It can't be read, right? So and it will hamper the process of protein formation or protein synthesis, right? So that's why this mRNA is found generally in a single structure, in a straight manner. Understood? It is, that is the primary structure of RNA, not secondary structure. Got it? So your answer is false. Every RNA, it doesn't have to be a secondary structure. If this mRNA structure becomes changed to primary to secondary structure, then it will hamper the translation process where the protein cannot be synthesized. Understood? So this RNA secondary structure depends on the order of base pairs. Almost all naturally occurring RNA is selected for function which depends on secondary structure, which depends on the base pair order. So it depends, likely the most stable secondary form of RNA involves the double helix. Fine. Generally the double helix form of RNA have been used as the different catalytic processes, but this mRNA, that is the messenger RNA, it needs to be in the primary structure to help in the translation process. Understood? I guess it's clear why it is false, not true, because it is written every, not all. Okay, not few. That means, I'm sorry. Fine. Then let's move to next question. In the splicing mechanism, which one is removed? Intron, exon, RNA or pre-mRNA? I believe this is quite simple question. Because we have just discussed it just few minutes before. Yes, very good. I'm getting good answers. Everyone, please try to answer. So splicing mechanism, it is a part of which process? Transcription, translation, replication, reverse transcription. Very good. It is a part of transcription. Okay, others please try to answer. I believe this is quite simple question to be answered. Okay. So as you have answered, the question answer is intron, right? This is the non-coding sequence. So this is the simple or brief explanation of your splicing mechanism. For example, this is your gene which has exon 1, exon 2, exon 3, and these are the intron parts. Okay. So when the start of transcription, after this transcription, this is called primary transcript, and after the splicing mechanism, the introns have been removed, only the exons are placed like exon 1, 2, 3. And after this intron is removed, these two exons have been ligated, okay, by the enzyme ligase. This is very simple briefly explained. There are lot of enzymes and other things also required for the whole mechanism of splicing. You can read about it from the specific book or any article or any journal also. So this is the last question of today's session. Please try to answer. Which one is not a feature of hammerhead ribozyme? So this is one example of your small ribozyme, that is hammerhead ribozyme. We have talked about RNase P, that is ribonuclease P, which is an example of your this and also self-splicing introns. These two ribozymes we have talked about now, and also hairpin ribozyme, hammerhead ribozyme, and HDB. These are included in small ribozymes. So, which is not a feature of hammerhead ribozyme? Helps in virus replication, found in mammals, used for RNA cleavage. Next D is example of satellite RNA. Okay, then let's discuss. Most of you have answered C, used for RNA cleavage. Hammerhead ribozyme is a RNA molecule that catalyzes reversible cleavage and joining reactions at a specific site within an RNA molecule. Okay, it is used for reversible cleavage. It is a model used for targeted RNA cleavage experiments with potential therapeutic applications. Okay, targeted RNA cleavage. Next is named after hammerhead shark due to molecular resemblance. Hammerhead ribozymes RNAs were originally discovered in two classes of plant viruses like RNAs, satellite RNAs and virions, that is virions. They are been found to be widely dispersed within many forms of life like insects, plants and mammals. That means it has been found in lots of like different range of organisms including viruses to mammals. And generally it is a model used for targeted RNA cleavage experiments. Understood? So see which one is wrong about it. Not a feature. It says it is not a feature of hammerhead ribozyme. Example of satellite RNA, yes, it is true. It is written here that it is an example of satellite RNAs. That means, कहाँ गया? So it won't be the answer. Used for RNA cleavage. Yes, it is used for RNA cleavage in specific experiments. Found in mammals. Yes, it is found in mammals. So which one is not a feature of hammerhead ribozyme? So helps in virus replication. Which ribozyme helps in virus replication? HTV, hepatitis delta virus. Understood? This HTV ribozyme is useful in this virus replication process. Okay, so now we have discussed about different forms of ribozymes. So basically the today's session was about RNA as enzymes, that means ribozymes, and there are two class of ribozymes, where one is metalloribozymes, which includes your ribonuclease P and self-splicing introns. Next one is your small ribozymes, which includes hammerhead ribozymes, hairpin ribozymes, and HTB, that is hepatitis delta virus ribozymes. Understood? So we have discussed about all the mechanism of these five different ribozymes, which is how what is the structure of these ribozymes and how this been helpful in different types of metabolic processes and all. So if you have any doubt regarding today's session, then please write the question in the chat box. I'll try to clear it. Do you have any question? Okay then. We'll see, we'll meet again in next Monday, on next Monday, where we'll have the same class and we'll discuss about the different topics about RNA biology and some questions also. Okay. See you then. Thank you for joining.